So there are a set of practice problems. I believe those are only 3 problems. They're very simple. There's 2 monohybrid crosses and 1 dihybrid, or maybe they're all 3 monohybrid. I'll look through my, I have physical copies of them. I'll look through right now and verify that with you. So there will be practice problems, and we're going to go through a couple of those today. And then there are also what I would have titled as homework genetics problems. And I'm going to assign the spring set. So if you've done the fall set, great, good for you, right? Or if you have questions about the fall set, great, good for you. They're the same problems. I just changed the topics, right? So in the Halloween problems or the fall problems, there's a problem about witches with stringy hair, and speaking of stringy hair and scraggly pointy fingernails. And then in the spring problems, there are tulips that have 2 colors, or a color and a stripe. But it's the same, you set it up exactly the same. If it's a dihybrid problem, it's the same problem. It's just with a different theme, okay? So it's not gonna hurt you, or I don't wanna say hurt you. It's only gonna benefit you if you do all of them. I get it. Most of you don't have time to do all of them. The spring set is the one that I'll put in as an assignment, and I won't have it due until probably Friday, so that you have time to look at it and complete it either before or after the quiz. That's fine with me as well. So let's take a look at those practice problems, because I think that'll help us figure out sort of how to set up the problems, and we'll just go through it again. I know that we did a few of these before spring break, but I realized most of you were still focused on getting ready for the mitosis and meiosis quiz. So I'm looking, like I said, I'm looking through my physical copies right now to see. These, I believe, were the practice problems. Yeah, and there's a correction I have to make to the spring problems. I accidentally wrote the wrong color. I put white stripes and it was supposed to be yellow petals. So I'll fix that. And then, so there were a set of 3, actually there's 4, they were titled practice problems. And the first one was about peas. The second one was about peas. The third one was also about peas, and the fourth one was also about peas. So these were all Mendel problems, and they mimicked the problems that were probably, I believe, in your textbook that were actually written out problems. So I thought it would be a good place to start, because it would help you with the vocabulary and help you remember the words to use and how to turn the words into a Punnett square. I would remind you that I've also posted all of those note sets are really important for this particular sets to help you with the problems, particularly the one that says how to translate genetics word problems into Punnett squares. Okay, so I would really advise that you, if possible, print those so that you have them next to you while you're trying to set up the problems. So let's take a look at these, what I had originally titled practice problems. And I'm going to just give us a whiteboard here. I'm going to hide everyone, and I'm just going to kind of start. And if you guys have questions, of course, please just interrupt me. I won't be checking the chat because I'm going to try to work through one whole problem at a time. So let's just start with the homework problem 1. Okay, and the homework problem 1 was titled, this is, I'll read the problem, I'll try to read it slowly. In peas, round seed shape is a dominant allele to wrinkled seed shape. Homozygous for round was crossed, that means was fertilized, with a plant homozygous for wrinkled seeds. Draw a Punnett square to determine the phenotypes and genotypes of the F1 generation. Draw a second Punnett square to determine the phenotypes, which means appearances, and their proportions, meaning the fractions, or sometimes you'll see that called percentages, for the F2 generation. excuse me. So I just want to real quickly remind you, and I used all of those vocabulary words in that first problem on purpose so that you would be able to sort of process this as quickly as possible. So I might change my mind here and set this as a just on my chalkboard because we'll see here. All right, so let's get a start, right? So in this particular problem, in problem number 1, right? So it says in peas, round seed shape is the dominant allele to wrinkled. And in this particular problem, I actually gave you the letters to use to designate round and wrinkled. So remember, the dominant allele, that tells us a couple of things, right? That tells us, we always see that in the phenotype of the F1 generation, phenotype of F1, meaning what the F1 phenotype, physical appearance, excuse me, what it looks like. So they're telling you the dominant allele for seed shape, allele is the version, right? The trait or character in this case is seed shape. Okay. And for each character in these first problems, we have 2 choices, right? We have a dominant choice and a recessive choice. We have 2 alleles. And again, they're already telling you that in this case, the dominant allele is round. So we use the first letter of the word round and we make it capital. And we use that for our symbol to designate the alleles that are in the gametes of this particular plant. Now we have a dominant allele pea plant with round seeds. And we are going to cross that plant with one that is wrinkled, right? We do not ever change the designation. We keep using the same letter in peas. We use lowercase r. And the only time we can ever see wrinkled, which is the other phenotype, the only time we can ever see that is if we have 2, because it's recessive, is if we have 2 copies of wrinkled. Now it's telling us in the problem, that was just defining the alleles. Maybe I should erase that and start with, let's just leave it at this. Round seeds is dominant and wrinkled is the recessive. I find it helps sometimes if I do this, if I put round with a blank, meaning round with either of the other alleles, either another capital R, if it's homozygous, or the heterozygous, if it has lowercase r, either one of those is going to give me round. But the only way, again, I can see wrinkled if it's the recessive allele is if I have 2 copies of wrinkled. So this blank line next to this capital R designates this. We have either this or capital R, lowercase r. Okay, so that's to start with. And I find it a good habit to always write that down at the beginning of the problem, because then I don't have to think too much about that when I'm actually trying to set up the cross and draw the Punnett square if I have that written out. So I have my character, seed shape, and my 2 alleles, the round allele and the recessive, and it told me in the problem that the round allele was dominant, so that's why I gave it the capital R. And then the wrinkled allele is lowercase r. Okay, one plant that is homozygous for round seeds was crossed with a plant homozygous for wrinkled seeds. So again, I would remind you that homozygous refers to having 2 copies of the same allele. Okay, so 2 copies of the same allele. So if a plant is homozygous for round, then I would designate it like this. That means homozygous for round, crossed with a plant that is homozygous for recessive, two lowercase r's. Now it says, draw a Punnett square to determine the phenotypes and genotypes of the F1 generation. Remember, the F1 generation, that stands for first filial generation, or the babies, the offspring of a cross of two true breeders, or what we would refer to as P, parent generation, which are always the parents. If we're starting with true breeders, we call the parents true breeders. They're always true breeders, means two copies, means homozygous. So we need to know the phenotypes of the offspring, and that means what they look like. So that's usually not very scientific to say, what does that look like? We use the word, what is the appearance? What is the phenotype? So what do these offspring look like? And what is their genotype? The genotype, remember, is the alleles that are present. So now it asks us to draw a Punnett square. We would refer to our handout that says the first thing we have to do for our Punnett square, one of the first things is to determine my pages here, sorry. We have to determine which is dominant and recessive, check. Assign symbols to the dominant recessive alleles, check. Determine the parents of the problem. Okay, we did that, check. Draw the Punnett square. Okay, now before we start drawing the Punnett square, we need to say one more thing. We need to say, how many characters are we studying? How many traits or characters are we looking at? We are only looking at seed shape, one trait. So because it is one trait, we refer to that as a monohybrid cross. And that means we do a Punnett square with 4 boxes. Okay. So we draw our Punnett square. We put 4 sub boxes inside the Punnett square. We draw this diagonal line here and block it out to represent which parent is the female parent and which parent is the male parent. And in plants, it's not going to matter because they're flowers. And so they have both pollen and ovules. They have both male and female. It doesn't matter which one of these I choose to be the male gametes or the female gametes. Okay. So the female gametes, I'm just going to pick one, right? So I'm going to pick this one and I'm going to put a blue circle around it just to represent. And sometimes you'll see if you watch the videos, sometimes you'll see, I'll just write a circle to remind us that we're talking about this particular parent. Okay. So I'm going to put the blue circle on there and I'm going to say, okay, what possible gametes can this parent give to the offspring? What possible gametes can be put into in this case, since it's the female, it's going to be into the ovule. And I only have one choice. I have capital R or the other capital R. Why do we do this? Why use both of them? Because as we're going to see in the next chapters, even among alleles that are technically almost identical or give us the exact same phenotype, there could be a mutation. There could be a slight variation or a slight difference. And we're taking that into account by putting both of those R's there. Okay. The other parent, and I'm going to put a triangle around that parent because I usually do. And I would put a triangle here if it didn't matter which parent was male or which parent was female. And I'm going to put the lowercase r's, one above this box and one above this box. And I think we highlighted this 2 weeks ago when we last had this conversation, as a reminder that these here, and I'm going to put circles around these and triangles around these are gametes. These are alleles in, I should say, a gamete, right? These are the alleles that are in a gamete. And the point of the Punnett square is to try to figure out all the possible combinations of gametes. If we only have one trait and we're only doing a monohybrid cross, there isn't a lot of variety or difference, but we start simple and we build to more complicated. Okay, so just be patient. So now we fill in the Punnett square. That's the next step, right? So it says I'm fill in the Punnett square. I think that's step 5 or 5 or 6, right? Draw the Punnett square, mate them, right? And then that means fill them in and give the zygotes and write the proportions to the right of the Punnett square. Reread the problem and make sure that you answered all the parts of the question, right? Okay, so let's fill in the Punnett square. So we would make this capital R and then the lowercase r, and again, capital R. We always put the capital letter first because that tells us what the phenotype is going to be and helps us find it very quickly. All right, so we put what happens if this ovule gets fertilized by this pollen grain is what we're figuring out. What zygote will we have? We will have this zygote because it's the combination of these two. Now, the question asked us, draw a Punnett square and determine the genotypes and phenotypes of the F1 generation. These are the F1 generation right here. The zygotes are the F1 generation. So to the right of the Punnett square, we would write all are round because that's the phenotype, and all the genotypes are capital R, lowercase r, right? And if it helps you, you can write phenotype next to that and genotype. I would encourage you to do that just for practice until it becomes second nature. The tricky bit is now they're going to ask you for the F2 generation. So I'm going to add another whiteboard here. Oh crap, I was sharing, right? I think oh, there, it's still there. Is it still there? Let's go. Okay, so anyway, let's add another whiteboard here and go to the F2 generation. So the F2 generation is 2 F1s crossed with each other. And how I know what the genotype of the F1 is going to be is based on, I'm going to draw the other Punnett square we had over here in miniature, right? We had capital R, lowercase r. Big R, little r, right? Like that. So all of these, remember in yellow, I had written that these were the F1s. So now I'm going to take one of these F1s and let it, in plants, you can let it self-fertilize. If it was a fruit fly, you'd have to mate it. You'd have to pick one of them as a male and then pick another one that was a female. So you'd mate brothers and sisters together, essentially, right? But you cross 2 F1s with each other, or you let them, we say, self-fertilize if it's a plant. Then you're going to draw a second Punnett square. And we're going to put the letters on top of that. All right, I'll put a blue circle around this one, right? So we'll do it the same. And a green triangle around that guy, right? And this would be the green triangle. So this parent, circle parent, can give either a capital R in its pollen grain or a lowercase r in its pollen grain. And the triangle parent can give either a capital R in the ovule or a lowercase r. Because remember, the gametes are N. They only have one copy. The zygotes, those are diploid. They have 2 copies of each characteristic, each allele, 2 copies of alleles for a characteristic or trait. So now when we put these alleles together, capitals first, lowercase second, and then to the side of this, again, to the right side now, we would be writing out what are the phenotypes, because the question asks us, draw a second Punnett square to determine the F2s, the phenotypes and their proportions, meaning the genotypes, right? Or sorry, the phenotypes and the proportions. So how many of each phenotype? So we start with this one. What does this one look like? That one's round, right? What does this one look like? That one's also round, because remember, capital R anything equals round in the phenotype. The only time we see wrinkled is when we have two lowercase letters, right? So that one's round, this one, round. And see how I'm crossing them off, because then that helps me not count them twice. And then this one is not round, this one's a wrinkled. So I have 3 round and 1 wrinkled, and we express that as 3/4. 3 out of the 4 are round and 1/4 wrinkled. And that would be the complete answer to that particular question. So I'm going to pause and ask if you guys have questions about that before I go on to doing the tall and dwarf. Questions, comments, worries, concerns. Where are you guys? Where's my gallery?
So I have a question. So when
Sure.
you answer when you write the answer for the genotype and the phenotype, you just put the homogeneous and like for the phenotype of a proportion, you have to put 3/4 or
You have to write 3/4. Yes.
that's
So you have to write 3/4 because I think I lost all of my, let's see if my whiteboards are still there. No, they're gone. Okay. Let's do this. Whoops. Let's stop the share and switch to make it me. Switch the camera.
Would my notebook help you at all?
Yes, that would help. I think I'm just going to, I think I'm going to put the camera up on the chalkboard to try it a little bit up on the chalkboard. But I haven't done that in a while, so I have to kind of reset up the room here. That's a little better. All right, I'm going to make you guys go away. So when you have your Punnett square, that looks pretty good. All right. So when you have this, and you have it all filled in, you must write to the side of that 3 out of the 4 are round and 1 out of the 4 are wrinkled. That is telling you, that's the complete answer to the question. That is telling you, oh, it's all backwards. Forgot about that. That's a Zoom thing. Crap. That's that's telling you that the 3/4 and the 1/4 are telling both the amount of the phenotype in question as well as the phenotype. If I do that, no, that's a Zoom thing. Let me try to fix that. So yes, you have to include both. Yeah, I forgot. I haven't ever switched it on this version of Zoom that I have now. I didn't switch the camera, rotate it 90 degrees. So I'm trying to find that while we're in process here, and I'm not getting all of my choices, all of my let's try backgrounds and filters. That's not it. All right, well, I'll try to fix that for tomorrow so that we can do this again. That's not going to work, so we'll just stick with the whiteboard then.
The chalkboard looked fine to me. It
It
wasn't
was
that.
first. It wasn't backwards.
No.
Oh, well, that's good. I don't know why it looked backwards to me, but yeah, it's reversed for me. That's odd. Okay, well, if it's good for you, then I'll keep using the chalkboard. It's a little bit easier than the whiteboard for me. So the second problem, I'd like you guys to try a little bit on your own. If you wouldn't mind, I'd like you to try to do that second problem, the one about studying the character for height in pea plants. I'd like you to try to do that one all by yourself and see what you come up with. So I'm going to give you a minute. I'm going to erase my peas here. These peas, so that we can put the other peas on the board. Again, I've worded the problems on purpose to try to get you to use all of the vocabulary. So the second problem is about height, and it talks about true breeders. And again, it asks for which allele is dominant, which is recessive. Draw a Punnett square, show me the F1 and show me the F2. You'll start to see, I hope, that all the problems are basically the same, right? So the main difference is whether or not the problem starts with true breeders, which, remember, is also called the P generation, which is also always homozygous. Right? You also always need to focus on, right? You'll see the dominant allele, or if there's two traits, alleles in the F1 generation. And remember that to see the recessive allele in the phenotype, you need to have two copies of that allele. The dominant allele is going to be the capital letter. The recessive allele is going to be the lowercase letter. When it's one trait, the Punnett square only has 4 boxes. So again, where to begin, right? So the problem starts with he's studying the characteristic of height. So we know that the gene that we're looking at is the gene for height. And he produced true breeders for 2 versions or variations for 2 alleles of plant height, tall and dwarf. We don't know which one is dominant and recessive yet until they're crossed and we see what the F1 generation is, what it looks like. Because in this problem, you weren't told specifically which allele was dominant and which was recessive. So if you read the next sentence, he says, when he crossed a true breeding tall plant with a true breeding dwarf, all the F1 generation plants were tall. If all the F1 are tall, that means tall is the dominant allele. So then we choose T for tall. And remember, if it's a true breeder, we're going to have two capital T's. Homozygous. Dwarf is going to be the recessive allele. And a recessive true breeding dwarf plant would then be designated lowercase t, lowercase t. So I'm going to have basically the exact same Punnett square again. We had for round and wrinkled. F1 generation, everybody's tall and everybody is capital T, lowercase t, or heterozygous. Remember, these are the F1s. So when we cross an F1 with an F1 to get us to our F2 generation, we would be crossing an F1, capital T, little t, that's its genotype, with another F1. And in this case, we would have 3 out of 4 would be tall because our genotype, capital T, or capital T, little t equals tall. And we would have 1/4 dwarf. And if it asked for that genotype, it would be lowercase t, lowercase t. Questions, comments, worries about that one?
This
Before we go.
might be a silly question, but
No.
does I get T tall, is that something to be hung up on to make sure I'm using the correct letter?
A little bit.
Okay.
So in Peas and in Mendel, we all use the same letters. Whenever you study the genetics of a particular organism, someone will probably have already designated what the letters or designation is going to be for that organism. And then you are expected to follow that designation, not invent new ones because when you're doing the same research or you're researching the same organism, and then you're going to present that material, everyone's expecting that we're all using the same words, right? It's no different than, identifying a human as Homo sapiens, genus and species, so that we all know we're talking about Homo sapiens when we say Homo sapiens. So yes, that is important. You can't change the letters, or you shouldn't change the letters. And as I mentioned, so like if you were studying corn, for example, a lot of the traits in corn are 2-letter designations because a lot of the traits begin with the same letters, like sugary and starchy, for example, or sugary and sweet, or sweet and starchy. There's lots of different designations. And so the only way to keep them straight is to use the vocabulary of that particular organism. It changes when you get to chapter 15 because they introduce fruit flies, and the designation in fruit flies is different. We use capitals and lowercase letters. However, in fruit flies, we also designate the wild type with that plus sign. So when you look at some of the problems from chapter 15, for example, if you read that first part of chapter 15, I think it's 15, right? About the sex-linked traits, right? So if we're looking at a sex-linked trait, and in particular, if we were studying fruit flies, right, we designate the traits still the dominant allele with a capital and the recessive allele for a trait with lowercase. Sometimes they might be 2-letter designations. However, what's different about when we start talking particularly about animals in general, we use a plus sign to indicate the wild type allele, which means the one that is the most prevalent in the population. And sometimes that happens to be a recessive allele, and sometimes it's not. Sometimes it's dominant allele. So we designate wild type as a plus, and then the mutant or the mutation as the letter designation, right? So for example, like if we're talking about a red-eyed fly versus a white-eyed fly, red eyes is the wild type allele. So if you have a red-eyed female, a homozygous red-eyed female, she would be designated like that. And I'm going to put a little circle around the X so it doesn't look like one of the sex chromosomes. And then if we had a white-eyed male, right? Trying to make that look like a lowercase w, because white eyes happens to be a recessive mutation. And you're thinking, well, why don't you have 2 little w's? Because this is a male fly, it only has 1 X chromosome, and the trait for eye color in fruit flies is on the X chromosome. So I can't give that male fly 2 little w's because he doesn't have another X to put the W on. He has a Y instead. This complicates things if you're looking at those problems from chapter 5, as well as when you attempt the sex-linked problems. I believe there's a problem for sex-linked trait in both the spring set and in the fall sets. I believe it's number 4. It's the chickens with a multi-feathered tail in the spring set, and in the fall set, I think it's being a werewolf. Anyway, let's stick with the fruit flies for now. So when we would construct this Punnett square, we would now be very careful where we put the male and the female flies, because we're not only keeping track of the traits, the white-eyed trait, but we're also keeping track of the sexes. So this fly would be a female fly that was carrying the trait. This would be a female fly that was carrying the trait. This would be a red-eyed male. And this would also be a red-eyed male. So you can think of this like an F1, right? Sort of like an F1. And we have all red eyes, right? So it still follows Mendel's rules. Wild type in this case is dominant to the white-eyed being recessive. So it's still, everybody has red eyes, whether they're male or female. It's not until the F2 generation that we see some differences. And I'm going to just erase this part so I have a little bit of space to draw the second Punnett square on the same whiteboard for you. Okay. So in this case, my F2 generation, so if I crossed one of these males, doesn't matter which one, with one of these, one with its sister, right? Doesn't matter which one, they have the same genotype. So the genotypes are going to be, I have a female carrier crossed with a wild type male. All right, so let's look what happens when we do that. So female carrier for white eyes crossed with a red-eyed male. And again, to help me remember, like if I was doing this for the first time, I would keep it over here. Plus equals red eyes, right? It's the dominant allele, it's wild type. Little w is white-eyed flies, right? I would just, there's no shame in writing that out to the side so that you can remember it. It's not important that you have that memorized and at the tip of your fingers all the time. All right, so let's take a look at our flies then. So this is going to be a female fly because it has 2 X's, and she is homozygous for red eyes. This is a carrier female because she's going to have red eyes, but she's carrying that mutation for white eyes. this would be a red-eyed male. Look here. Here we end up with our white-eyed male. So it's still, if you're looking at just the characteristic, we still have 3/4 have red eyes and 1/4 have white eyes, right? It is just that of these 3/4, these are some males and all the females have red eyes, and the 1/4 white eyes only in the males. I won't be asking you for that much information on the sex-linked characteristic or a sex-linked problem. I would probably just be asking you, what is the probability that you would get a white-eyed male? And you would have to tell me 1/4 and show me the Punnett square. Come to genetics, you can learn more about it, right? Questions, comments, worries about that? All right, then, I'm just going to leave that there. I'm going to ask if anybody has any questions about anything else, because I realize that although a couple of you came specifically for the genetics problems, some of you may have come for something else. So if anybody has any questions that are outside of the genetics problems, I want to give you an opportunity to answer, to ask those now and have my answer before, because we're getting close to 10 o'clock, before I stop the recording. And then during my office hour, again, everybody's welcome to come back and we'll do more genetics problems if you want to, or not. We can do something else, depends on what you need. But if you have just this hour to talk to me, I wanted to give you a chance to ask me other questions if you had any other questions that were pressing on your mind before I just kept blabbering on about genetics problems. Yes, no? You can just unmute yourself and ask.
What do you know about the chapter 15?
Sorry, Bo, I can't hear you very well. Can you ask me that again?
The note is more about the chapter 14. So what do we need to know about the chapter 15?
Yes, there should also be notes for chapter 15, very brief notes about chapter 15. I believe the focus is entirely on just those determining sex and how to do a sex-linked problem. So I'm looking through my things here. I don't see it jumping out at me. Maybe not. How about this? I'll look into that and I'll try to get back to everybody today on an announcement or something about chapter 15. I'm not sure what is still, or quite honestly, I'm not sure what is still in that quiz. I would hope that it included sex determination, like male and female, XY, XX and XY, and then a sex-linked problem, but I'm not sure. I don't think there's a sex-linked problem on the exam, but I'm not positive on the lecture quiz 5. I'm not positive. I'll have to check on that and get back to you. Where's my list? Quiz on Wednesday. So that gives us both today, office hours today, office hours tomorrow, as well as Wednesday for you guys to ask more questions about the genetics problems. And I'm making myself a note right now. I will look to see if any chapter 15 on the quiz. And I will put an asterisk star next to that. I wrote it in red pen and then post as an announcement. And I will try to do that today, so that you know whether or not you have to focus on that. Like I said, my suspicion is it's very minimum. It's just sex determination, meaning XX are males, or XX are females, and XY are males, and how to determine a sex-linked trait. There are not very many sex-linked traits in humans, but some of them have very profound effects. So hemophilia is a sex-linked trait. So that can obviously affect blood clotting and wound healing for people. And so that would be one that would be important if you knew that you had it or not. Most other conditions in humans, it's hemophilia, Duchenne's muscular dystrophy, and colorblindness are sex-linked traits and common sex-linked traits in humans. Most of the rest of our genes are carried on our other 22 chromosomes, not on our sex chromosomes, just a few. So that's kind of important for you to know. And many traits are simple Mendelian traits. So I would want to say that many of the characteristics like that we've already spoken about, some characteristics have that added sort of, I don't know, maybe I would call it an added layer of, they might be an incompletely dominant trait. You would know that if the trait, if a third phenotype, one way to determine that is if a third phenotype appears. When you cross two true breeders, the example is always flower color in snapdragons. If we cross a true breeding red flower with a true breeding white flower, you actually get all the F1 are pink. So that's an example of incomplete dominance. And it means just like it sounds, the red allele is not completely dominant over white. It can't completely shut down the production of the pigment. So you get this in between phenotype, which we would just designate the heterozygote being pink, right? And in this case, to get a red flower, you need 2 capital R's, and to get a white flower, you would need 2 little r's. The other added layer that we bring up in chapter 14 is multiple alleles. That's important because you need to know a little bit about that for blood groups in humans, which I'll probably spend a little time talking about more about that on Wednesday, because that relates to your lab that you're doing, that ABO blood grouping lab, blood typing lab. Multiple alleles means more than 2 for a particular trait or characteristic. And blood groups in humans is a good example, because in humans we have 3 alleles for blood type. And it's extra complicated, the blood group, because it's not only multiple alleles, but allele A and B show co-dominance. So notice how I wrote both of them as capital letters, because they are both dominant alleles. So there's 3 alleles. We designate them with a capital I and a capital A superscript for A, capital B superscript for the B allele, and then 2 lowercase i's for blood type O. Which I understand is a little confusing, but those are the technical terms for it, and that will come back to haunt you in anatomy and physiology. So that portion of chapter 15, or sorry, of chapter 14 will be important to focus on to help you with the lab activity. And again, I think that lab is due this Friday, if it wasn't due last. And it has a little bit of Punnett square work in it as well. Okay, any other questions that you guys have on your minds?